Okay. All right, so we are welcoming a very close friend of mine and one of my very first students. He actually wrote a poem about it. This is Stefan Roberts. If you guys saw the cooking video, he went by Devin in the cooking video.
I did what?
You went by Devin. So when he was,
I was his first student, and he tutored me in when I was in college, and he didn't know my name for the first like 6 or 8 weeks that he was my student. So he always called me you, until he finally got the nerve to call me Devin. And
I was wrong.
confusing, so
You'd think I'd look at the roster, but I was too busy. So and then we became fast friends. Maybe it was because I felt guilty. I don't know. Anyway, so Stefan's doing really, really cool work at Duke University. And he's going to share some of that. This year, he can't share exactly the work he's doing because a lot of it is not yet published. But he tells me in the next couple of years, he'll be able to come back and tell us more about that specifically. But I'll let him explain everything. Just a reminder, anytime you're asking questions, we have a camera trained for questions, but you need to make sure you're using a mic. I don't have the speaker set up, but it is all being recorded so that it'll be good quality for the lunch and learns. We also have a new thing. We're recording the PowerPoint presentation as it goes. So in the corner of the video, we'll actually see the PowerPoint, whatever slide he was on at any given point. Cool? Probably around the time you switch the first slide, say something like switch as you're switching it, and that'll allow me to line it. Does that make sense? Because I'll be able to put a marker on exactly where the first slide switch is.
Okay. You want me to say it every slide?
No, just the first one.
Okay.
It's fine. All right.
All right, cool. So, obviously, my name is Stefan. I'm currently a PhD student at Duke, and I'm going to talk a little bit about, well, I'll say proteins as materials, and we'll get into what that means as this, I guess, talk goes on. But it's a little bit of a general overview of maybe a different way of thinking about proteins and not thinking about them as enzymes, but actually thinking about them as functional materials that we make using a variety of biotechnology techniques that Avi's been telling me that you guys have been learning. I should also say, please interrupt me. I don't want to go this entire time without being interrupted. And I get really uncomfortable if people don't interrupt me, so please ask me as many questions as you feel is necessary. And it doesn't even have to be about what I'm talking about. I've known Avi for almost a decade. If you're just curious what he was like 10 years ago, ask me questions about that. Yeah, all right, so a little bit of back history. I did a lot of dabbling before I landed on the work that I'm currently doing. So when I was in high school and for the first year or so in college, I worked at and with some researchers at St. Jude Children's Research Hospital on projects related to determining which genes are responsible for pediatric cancers. They tend to be different from genes that are associated with adult cancers because they tend to be mutations that the children are born with or that they adopt very early in life. And so it's usually a very simple point mutation, a very simple frame shift, something that can be identified and cured more easily than, say, a lot of the mutations that you get in adulthood where you've gotten thousands over your lifetime and nobody's really sure exactly what is causing the cancer. I also worked on a couple projects that had to do with fungal pneumonia. The children that go to St. Jude because they are under treatment for cancer therapy, they have very low immune systems. So St. Jude actually has a really interesting infectious disease department because they see things that nobody else in the world really ever sees. So after that, I went to college at Washington University in St. Louis, which is where I met Avi. And I did research there on metallic nanoparticles and using them for two different types of things. One is just for determination of shelf life. We made very oddly shaped particles that had interesting properties when you shined light on them. So you could put them in a solution with a drug and you could shine a light through that solution. And then based on the properties coming through, you could tell how long or how degraded the drug in that solution was. And the other thing that we used them for is something called radial thermal ablation, which is super fancy, so I apologize. But it's the idea that when you get a radiation treatment for cancer, that heats up the tissue in the area. And so if you put metal nanoparticles there, it heats up the metal nanoparticles and they ablate and kill all the cells. So it's like a dual targeting system for treating cancer. And now I'm at Duke. I'm in my 5th year of my PhD. So I got about another year. And And I'm working on what we call bio-inspired materials. So these protein-based materials. And specifically, my research delves into their use for tissue engineering. Like Avi said, that work is not published, and also we have some IP pending, so I can't really talk about that. But I can talk about what our lab uses them for in general, and then specifically for some drug delivery concepts. Super random things about me. I have an extremely extensive collection of vintage Pez dispensers. That's kind of a fun fact. I was, at least for a little while, a semi-professional disc golf player. Also fun. My sport love is soccer, though, so I also play soccer. And then I do landscape photography and stuff on the side. So this is a picture. I used Duke to send me to a conference in Switzerland last year, and I stayed there for a couple weeks afterwards. And so this is a picture from one of the sites that we went to and did some hiking in Switzerland.
Fun fact, interrupting you. All of our camera choices was based off of Stefan recommended the camera he uses to us as one. And so my fiance and I got that for our 5th anniversary for each other, and then I liked them, and so I bought them for our school programs.
All right, any questions about me so far? Yep. Oh yeah, that's a great question. So, do you see the one in the middle, the flower? It's one of the 2 psychedelic Pez's that they made, and they made them in the 70s, and they all have like the love and peace thing going on the side, but they have one that's a eyeball being held in a hand, and so I always liked that one. That, and because super nerd, they have one called an arithmetic, and there's little dials on the side. You can use it to do like basic multiplication tables and stuff. So that one also was one of my favorites as a kid. Anything else? Yep.
If you could pick any place to take a picture, like of landscapes and things, like where would you go?
Where would I go? I've been trying to go to Iceland for a couple years because I recently got to also go to Hawaii and do some shooting, and I was able to take some pictures of some volcanoes, and some of the pictures of volcanoes I've seen in Iceland are very intriguing to me. So I'd like to go shoot them. All right. Yeah. Okay. Okay, so we're gonna talk about materials and proteins as materials. Big question first. What do I mean by a material? What do I mean by a medical material? It's a super broad topic. It means like literally anything that you could put inside of a person. So that can be metal prosthetics. It can be heart stents or heart valves. In case you don't know what stents are, actually, does anybody know what a stent is? Yep. Yep, you're good.
Like a net-like thing that they put into veins or arteries where there's like clogging, so they like use it to like open up the vein or artery to like make blood flow better.
Yeah, that's exactly it. It's when you get a lot of plaque buildup inside your veins or arteries, and it's restricting blood flow. That could be a material you put in the body, or we're even starting to get to the point where we can 3D print scaffolds to actually grow tissues in. So the top left image here it's not really an ear. It's a plastic scaffold, and they're growing epithelial cells. So they're not growing any of the cartilage inside the ear, but it's a demonstration that you could theoretically grow stuff in the shape of tissues that currently exist in the body. Side note, I don't have this up there. The one tissue right now that we can actually 3D print and grow at will is the bladder, and it's because the bladder has very little vascularization. It's very thin, and vascularization is one of the main issues that's preventing us from just making tissues at will, because everything else is there. Yep, question.
What is that? What'd you say? Vaporization?
Oh, sorry. Yeah. Please. Yeah. Vascularization is just how many blood vessels get into the tissue.
Oh, okay.
Yeah. So you need a certain density of blood flow in order to get healthy tissue, and you need all of your tissue to be X number of microns or millimeters away from the nearest blood vessel. And so because the bladder is so thin, that's not an issue for that. Yeah, you keep me in check on jargon words. Okay, so that's just the material, but what about proteins as materials? So the idea behind protein materials is actually kind of elegant. It's the idea that nature has spent millions and millions of years evolving proteins for specific applications in the body. So why can't we just use those use those proteins and those materials that have already been developed and put them back, repurpose them for some sort of engineering method in the body. And this is just an examples of some materials that you may have heard of. Collagen. Collagen gets talked about a lot in the context of, I guess, beauty and facial structure. But collagen is an extremely important part of all of the extracellular matrix, which is just the stuff that your cells are sort of suspended in the body. Silk gets talked about a lot, and silk is one of the main materials that there is research on, because it has such crazy properties. So a quote that's often said is, if you took silk fibers and you made them as thick as a steel beam, the silk fibers would be 100 times stronger than the steel beam. So that sort of also gives you an example of why we might be interested in these proteins as materials, because they can have properties that we can't even get with non-biological systems. Okay, so in order to, I guess, think about proteins in a different light, think of them as materials, we got to take a step back and sort of redefine what a protein is. At its core, a protein is just a bunch of amino acids. You could even call it a polymer of amino acids. And it's often taught that proteins have a structure-function relationship. So those amino acids wrap into a secondary structure. Those secondary structures come together in tertiary structure, and the tertiary structures sometimes form together into globular structures. And that shape is what gives a protein its function. And the most common example of that would be an enzyme. So any enzyme you use is a protein that has a very particular shape. It's got a certain binding pocket, and that shape gives it its function. But in the last, 10 years really, it's really quite new. It's been discovered that almost 30% of the proteins in our body have almost zero or little to no structure at all, but they still have some sort of function. So we're moving from a structure-function paradigm to sort of a disorder-function paradigm. And it turns out that a lot of the proteins that have these disordered regions are are also proteins that are more mechanical materials in the body. So again, we're going to talk about silk for a second. One of the best examples of that is silk. So if you think of materials as being on an extreme of crystalline and extreme of just amorphous silly putty, silk is right in the middle, and it has these beta sheet domains that act as crystals, and that keeps the protein together. It keeps it interacting with the environment. But it also has a lot of sort of amorphous, destructured areas, and that's important because silk has to be able to stretch and relax. The only material in our body that doesn't really need to be malleable to some degree is bone. So as soon as you're outside of mineralization and sort of trace metal systems in our body, we've got to have some sort of malleability. And so including disorder into order allows you to have both stability and malleability. All right, I'm going to stop here. Check for any questions. Anything? Yeah, you look intrigued. Okay. Avi, do you have any questions?
Yes. So do you guys remember when we were talking about the proportions of structural components like beta pleated sheets and alpha helices, and we talked about globular and structural proteins? Remember, it had a lot to do with this kind of thing, and how the structure of the protein would correspond to its function, besides just the idea of an enzyme having an active site. The other thing I want to point out to you guys, what do you notice on this slide besides the images? Raise your hand. The hint is I'm always asking you guys to make sure to have this on your slides. Oh, you got it.
Is it the citations?
That's correct. So this morning when he was putting it together, he's like, shoot, I need another citation. I need the citation for this one. And I was like, oh, that's perfect. I'm always telling the kids they need their citations. So apparently that doesn't stop.
You need it later in life, because if you accidentally give a talk and the person who made that figure is in the audience and you don't put a citation, you get berated. And so giving, and because when you spend 6 years doing a PhD, you want everybody to know it was you that did that work. So that's why everybody's picky about them. All right. So protein materials, advantages over non-protein materials. So we'll say advantages over metals, advantages over plastics. One is biocompatibility, and this is actually one of the biggest ones. It's when you implant something in the body, you inject something into the body can have a response to that. It can have an inflammatory response. And that is often a very bad thing. It doesn't have to be a bad thing, but you definitely don't want your body rejecting what you're putting in, because then instead of helping the person, you're going to be introducing an entirely new different type of disease to them. So because proteins are from your body, they're intrinsically you don't have these issues with biocompatibility. The second is a little more complicated, but I'll call it genetic level control. And so if I say the word polymer throughout this, I'm really just meaning it's really just a plastic. So anything in your life that is a plastic is really just a chemical polymer. So we talk about proteins in my world as also being biological polymers. Not just because they're polymers of amino acids, but you can actually take repeat sequences of those amino acids and then repeat them, and then it's a sort of a 2-level polymer. So when you see biopolymer, it just means proteins, but just a very long protein. When you make a synthetic polymer, it's all chemically reactive. You have to mix in your chemical groups, you put them under certain conditions, you let that go for a certain amount of time, and then you get sort of a distribution of sizes for the length of that polymer. And let's say you want more than one different type of chemical group in there, you get a random but deterministically probable distribution of those chemical identities. When we're making a protein, we're using a lot of the recombinant techniques that I've been told you guys have learned. So we're making them at the gene level. So this all starts by making a gene. And because we're doing that, we know exactly where every single amino acid is. We know exactly where every single chemical group on every amino acid is. And we can modify those with like a molecular level precision, which is something that you absolutely do not get with chemical systems. And because we're making them at the gene, every protein comes out the same theoretically every single time. They're always monodispersed, same size, same distribution, everything. So biocompatibility and then this sort of genetic level control are why we're really interested in these materials. And then this is just a quick demonstration of the fact that we can actually take proteins and make things that look a lot like what you would see that you would make with plastic. So we can make these thin films, we can make sponges, nanofibers, hydrogels, things that have different properties such as optical properties, so whether they're clear, so you can actually do microscopy through them is a good example. Mechanical properties, you get the right protein, you play with it the right way, and you can make a screw out of it. So a screw out of protein is kind of an uncommon phenomenon, but a very, very possible one. So I guess this is just to show you that we're not limited as much as you might think by using something that has to be designed and actually produced by a gene. Okay, so specifically, the material that we use in lab is based on a protein called tropoelastin. So let me break down tropoelastin for a second. Elastin is one of the main components of what we call the extracellular matrix. That is the stuff is really the most scientific word that I can come up with. Stuff that your cells sit in and interact with in each other in their own environments. Elastin is particularly important in arteries and in your lungs and in your skin because it has the property of being able to expand and contract repeatedly billions of times without losing any of its function. Loss of elastin, by the way, is why as you age, you start to wrinkle. It's because you lose the elastin and you lose the collagen in your skin. And collagen, I should say, provides sort of the stiffness. Elastin provides the elasticity. So tropoelastin is like a mini version of elastin. You can think of elastin as the network. And yeah, Avi.
I'd like to be interrupted. Yeah. So you guys know aging's my thing. as well. So one of the reasons why, so he mentioned not just elastin but also the collagen. So when you visit old people, you often notice they have these sores all over their skin, and part of that has to do with their skin becomes really, really inelastic, but also tears a lot easier. And so that's a combination of the degradation of their elastin and collagen, and so their skin is not only not stretchy anymore, but also not held together very well. And so it's very easy for them to get bruises, cuts, etc. Because it's not just their skin, it's also the epithelial tissue and connective tissue underneath. That's why it's really bad for old people to fall, among other things.
All right, so tropoelastin also has that order-disorder thing that we were talking about earlier. It has these ordered sections that allow it to cross-link with other tropoelastin molecules, and that's where you get the elastin network coming from tropoelastin. And then its disorder nature in general is what it allows it to stretch and relax. So we use a protein that is, it's sort of a genetic minimal mimic of tropoelastin, and I'm not going to go into the details of exactly what it is. But the reason why we use it is because it has a very interesting property, and that property is sort of derived from its disorder behavior, and that property is that it undergoes a phase transition. So, anybody want to? I guess do you guys know what I mean when I say a phase transition? Anybody want to hazard a guess? Like a what? Not quite a change of state, but that's on par. So imagine taking water and then converting it into oil. That's sort of what I mean. So these are proteins suspended in a solution, and they're thermally responsive. So when you heat them, they no longer want to interact with water. They want to interact with themselves. So they then start to act like oil in water. And so what that looks like is this. So you're going to see this whole solution of protein go into a warm glass of water, and you see it immediately comes optically opaque. And then when you remove the warm glass of water and it's be able to cool down, it now becomes optically translucent again. So what's happening at a molecular or a microscopic level, if you can see this, is these little proteins are interacting with each other, and they're forming what looks like little oil droplet immersions. And so that's why it becomes optically dense, is because light can't penetrate those oil immersion droplets, those protein immersion droplets, and so you get a lot of light scattering. Now, why this is really interesting, makes it interesting as a material, is now you have something that you can treat like water outside of the body. But if you tune it so that it transitions when you inject it, it now can undergo this phase transition once it's in the body. And if you modify it right, you can actually change it. And I'll say this is what my research is on. So I don't have any figures about this, but I was able to take this polymer and make it such that instead of going from a liquid to a liquid inside the body, we made it go from a liquid to a solid. So now you can inject something as water, as a solution, and once it gets into the body, it now becomes a very solid, very mechanically stable scaffold. So how do we make these proteins? This is all biotech, all biomolecular engineering. Like I said, we design them at the genetic level, and then we produce them in E. coli. So we have an entire room in our lab that's devoted to just massive E. coli shakers.
Anyone have any guesses on why they produce them in E. coli? I can think of a few reasons.
Because it's like one of the most commonly used for like research. Like
That is a great reason. That's why people do a lot of things. But it's not what Avi's looking for.
So if they're already previous research, they can just use it because there's like less things that they don't know about it.
So one of my hints is he was saying that one of the reasons they like the idea of making a protein versus doing chemical processes is they know it's going to be the same every time. So why would E. coli be better for that specifically? So he mentioned that one of the nice things about making the proteins this way is that they can make them the same way every time. And so why?
It's asexual.
Okay, so we're not going to introduce new variation. So that's one part of it. What's a way that prokaryotes also in their production of protein have less variation in that protein than eukaryotes. What don't they do? I was looking for alternative splicing. They don't do alternative splicing. And so you're always transcribing and translating exactly what your transcript is.
So that is one of the reasons to use it. One of the other big ones is the life cycle of bacteria is very short, so we can get them to double every 30 minutes as opposed to mammalian cells, which take a much longer and much more expensive methods. We always joke in lab that if you walk, so we grow these E. coli in the vats of media, and we always joke in lab that you know that you're hungry, you know you need lunch when the media starts to smell good. So that's when it's time to leave. Okay, so we make these in E. coli, and we use that phase transition behavior to purify them. So we don't do traditional chromatography, traditional protein purification techniques. Because they become insoluble when you heat them and then soluble when you cool them, we heat them up, and then we'll centrifuge them down, and then the aggregates will be in the centrifuge pellet. And then we'll cool that down, resuspend it, and then centrifuge it again, and then the protein will be in the supernate. And we just cycle through that a few times until we're actually able to get a very, very pure protein just through simple heating and cooling cycles. And that also allows us to purify like orders of magnitude more than you can with chromatography. Okay, so we have these elastin-like polypeptides, these protein materials that undergo a soluble to insoluble transition. What can you use them for? So they've been used for a variety of things over the years, including sort of localized delivery. That one's really cool, actually. You can attach radionucleotides to the polymer, inject them directly into a tumor, and then that's something called brachytherapy. So rather than irradiating from the outside, you're actually irradiating from the inside of the tumor. But because these become insoluble when you inject them, they now form an insoluble mass that's not going to sort of seep out of the tumor. Tissue engineering, so these are elastin-based, right? And elastin is an important part of the extracellular matrix. So it logically follows that you could use these to grow cells, and that's very much been done. The intracellular phase separation, I'm going to mention this super briefly, and only because it's one of the most new things in my field. Papers on this have only been coming out in the last year or two, and it's the idea that these phase separations, this soluble to insoluble phase boundary, actually occurs inside cells, and that's one of the ways that we now know that cells regulate transcription of certain proteins. So these proteins can sequester RNA through a phase transition, and then inhibit its translation. But what I'm going to talk about now is they're used for two different uses as a drug delivery system. Oh, great. The first one is delivery to solid tumors using what we call a heat pump. And the idea is that if you inject this protein attached to a chemotherapy drug into the blood, and it won't transition at body temperature, but it'll transition just above body temperature. If you just let it circulate, it stays soluble. But if you actually heat up the tumor, which you can do using high-frequency ultrasound, for example, then you get an accumulation of your polymer and then subsequently an accumulation of your drug at the tumor. And so in this way, you're actually delivering the drug specifically to the tumor itself rather than just having disseminated chemotherapy drugs delivered throughout the body. Yep, question.
When you have to heat up that part of the body, how do you make sure that it's not damaging the tissues around?
Yeah, it's a good question. So there's a limit to the degree that you can go to. So you're medically allowed to heat the body up to 42 degrees Celsius without having to worry about damage. So when we do this in lab, we make these polymers transition between 37 and 42 because it can be that sharp. So that we don't have to go above that temperature. It's a good question. Any other questions? Nope. All right. Sweet. Okay. So the idea here then is if you accumulate the polymer and then you turn the heat off, now you have a huge amount of soluble drug that is directly near the tumor, and you have a concentration gradient. And that concentration gradient can then drive the drug into the tissue. And if you repeat this, so you heat, you collect polymer, and therefore collect drug near the tumor, and then turn off, and then you allow drug to disseminate into the tissue over and over again, you can get a really sort of positive tumoral response. And this is kind of cool, but this is how we first started looking at the ability to how local heating can change the aggregation properties of these polymers. We use something called a window chamber model. It's on a live mouse, and you put this little metal chamber on its we call it dorsal flap, and it keeps it thin enough that you can actually look through it with an optical microscope. So you can monitor the blood vessels and some of the tissue while the mouse is living. So this is a good example on the right. So this is our polymer ELP that's attached to a red dye, and so this is on a living mouse, and you're just looking into it, and you can actually see the blood vessels. So what that looks like with our polymer at low temperature, not that interesting. You just see, okay, so I should say, well, you'll see the difference in a second. But so you're just looking at the vasculature, new word that we learned, the blood vessels through this window chamber in a living mouse, and this is at low temperature. And then when you heat up, you can actually see the accumulation of the protein in the blood vessels. And we call this our protein lava flow. And so this is an idea of you're actually almost getting embolism where nothing else is able to go through. And then when you turn that heat off, it's now you can imagine what a high concentration of drug that you're going to have that you can get into the tissue. And so this is some real data from a paper a few years ago. This was showing that you need to do this cycling on and off multiple times to get an actual response. So with the black lines being heating and the white being cooling, if you just heat once and then you cool, you don't see a response. If you do it a few times, you don't get a response. But if you continue this heat cool cycle over about a 2-hour period of time, you can actually achieve a reduction in the size of the tumor. All right, and then really the one other way that we like to use these for drug delivery, instead of injecting intravenously or into the blood vessels, actually injecting, we call it subcutaneously, so just into the space underneath the skin. And we've used this before as a treatment for type 2 diabetes. You can attach drugs to the polymer, and in this case we're actually using a peptide drug, so we actually make the whole thing genetically. So it really actually increases the efficiency of our drug loading because we don't have to do a chemical reaction to get the drug attached to the polymer later. You can just produce the drug with the polymer and then make it and purify it. So we use a drug called GLP-1, and without going into too many details, GLP-1 is partly responsible for regulating insulin, and increased levels of GLP-1 will increase insulin production. So it's important for patients with type 2 diabetes that have insulin resistance or have difficulty producing insulin. And the idea behind this is if you inject something subcutaneously and you allow the body to degrade it over time, then you're getting a slow release of that drug over time, and you're getting a sustained release of that drug over time. That is opposed to having to inject GLP-1 once a day, once every few hours. You only have to do an injection once a week. And also kind of interesting, we designed these to be cleavable by proteases that are naturally occurring in the body. So protease, it's a fancy term for an enzyme that cuts a protein, which is actually most enzymes, but we put protease sites in between the different drugs, and so that's how we control how quickly it gets released, by how many of those protease sites we put in. And then this is just to, I guess, show that it works. So at the top, you can actually see, this was fluorescently labeled, and we were taking near-infrared images of the mouse. You can see the depot, which is injected here, sort of disappear over time as it gets degraded and as the drug gets released. And then this is just a measure of glucose levels. So in the mouse that has the depot injection, you have much more elevated glucose levels. It's a healthier mouse than the one that did not. So intravenous injection for the heat pump tumor response, and subcutaneous injection for sustained release of drug for treatment of diabetes. And I should mention that the last part, our lab has a company that is founded on the subcutaneous injections for the treatment of diabetes. And a couple of those studies have, a number of those have gone into phase 1 trials. This is an older slide, and a couple of those have even gone into phase 2 trials. Actually, quick question. Does anybody know the difference between phase 1, phase 2, and phase 3 clinical trials? Yep. I saw. Yeah. You're going to try, though, right? All right. So phase 1 is just to test whether it's going to be toxic. So you're almost not even looking for a treatment with a phase 1 clinical trial. You're just looking to make sure that what you put in the body isn't going to leave the patient worse off than you did before. Phase 2 is where you're looking for efficacy, and you're basically that it works. Phase 2 is that you're looking for the treatment that you do works well. And then phase 3 is where you're comparing that treatment to current state-of-the-art treatments to see if it's going to not just work, but work better than everything else that exists. So when something gets to, also money-wise, you need, let's say for you have a super cheap to produce recombinant protein attached to your drug for diabetes. For a phase 1 trial, you need like $10 million. For a phase 2 trial, you need $40 to $50 million. For a phase 3 trial, you're upwards of $200, $300 million. So people don't get to phase 2 and phase 3 very, very lightly because investors are very stingy with their money. So, all right, that's pretty much it. So we're going to start off the questions with a question that was already going.
Okay, so in like the drug making process, could you like if you find like a medicine, like do you patent it and then do you make money off of like it like once like, and who like funds the trials and everything, like who funds?
Yeah, okay, so there's like a bunch of questions in there, but I'll try to explain the process a little bit.
Okay.
If you are working for a company, most likely whatever you do belongs to the company. Nothing belongs to you. If you are working in academia and you come up with an idea, you come up with a new drug, you patent it, and you and whoever else worked on it and your advisor will have a certain share in how much ownership you have over that patent. And then the university will also have a share in how much it has. So, for example, the IP that we're currently filing, Duke has like 55 or 60%.
By the way, IP is intellectual property.
Yeah, sorry. So the patent that we're currently filing. Duke has about 50%. My boss has 30%, and then I have 20%. And then at any point in time, if anybody, like once you make it to market, you can sell your percentage, or you can just hold on to it and you negotiate with companies on whether you get royalties, whether they want to just buy the patent off you completely, whether you just, the most common thing is that you license it. So you still own the patent, but you let them use it for a certain purpose for a certain number of years, and they just pay you a lump sum money to be able to do that and for the exclusivity of being able to do that. Does that make sense? Cool. All right, so as Avi said, you can ask me about the lecture, or you can ask me about myself, you can ask me about Avi, you can ask me about anything. I'm just gonna sit up here with a microphone and hopefully I don't have to talk to myself. So.
Also to add, one of the things that I'm often telling you guys about is the skills you guys choose to gain and practice and work on here are ones that are then allow you to market yourself for other opportunities. And one interesting thing that Stefan has talked to me about, as I'm sure you're aware, his lab is one of the more prestigious labs nationwide and even worldwide in some ways, many ways, right?
In our field.
Yeah.
Let's say that.
And so let's say you were a student in biomedical or biochemical engineering. His would be the lab that you'd really want to be able to work in order to get that experience and that
sort of. I'm going to say a caveat here, and that you should also look for the research that you're interested in, because most universities you go to, a lot of the professors are very, very good. So people that are interested in materials tend to be attracted to our lab, but that's not always the case.
So one of the things he described to me was that he's very specific things he requires of anyone that would want to intern with him, and so. If you're curious about any of that, he'd be a good person to ask those questions to, because I feel like he's thought it out pretty well.
You had a question first. Yeah.
in general, going off of what Mr. Silber just said, what would you consider like a standout intern to be?
Yeah. Okay. So good question. So, I'm a little more picky than a lot of people. So I interviewed like 15 undergrads before I chose the last one that I decided. And so just for the interview process, I didn't care too much about. I didn't like. It's hard to care about whether you have research experience or not, depending on what year you are. So, if I was looking at a freshman or sophomore, I didn't expect them to have research experience. If I was looking at a junior or senior, I would wanted them to have some, at least some familiarity with lab, because I want to know that they can handle a pipette and like they know how to troubleshoot. Beyond that, I really just want them to be able to listen to me and formulate a couple good questions. It's and if they have done research experience, I definitely want to know that they know what they were doing. So that's one of the biggest filters. If you tell me that you've done something before and I ask you a question about it, that I feel like you should know, and you try to fumble through a response. Like in other words, just you know, don't ever get in a situation. This is a side note, a side rant. Don't ever get in a situation where a graduate student is telling you to do research and you're just doing it without questioning him. Because when people do that, they feel like they're getting lab experience, or feel like they're getting a lot of experience learning techniques, but they don't know why they're doing what they're doing. And so when they go talk to someone who wants to hire them to work for them and do those techniques again, if they didn't know why they were doing it, then that sort of eliminates a lot of their ability to troubleshoot. So, yeah.
Okay, so you were saying that they were using like proteins and stuff like to make like actual materials. So how could they use that for like nanotechnology and stuff? Like could you or like those materials like conductive? Like could you like put circuits on them and stuff?
So again, you're great at asking like 3 questions in one. So to address the nanotechnology first, there are 2 ways you can think about nanotechnology: nanostructures and nanoparticles. So fibers and networks and stuff are nanostructures, and because our proteins sort of tend to fold at a molecular scale anyways, a lot of these protein materials just natively have some degree of nanostructure. If you want to actually make a nanoparticle, that's a little bit more of an interesting question, and we can do that with proteins. We can actually do that with these, and that's another way that we use these. Remember, I mentioned that we can tune the phase behavior of these ELP polymers. So if you imagine you have 2, one with a temperature that transitions at say 60 and one that transitions at 20, and you attach them together, well then when you heat it up, it doesn't behave like a single polymer. It's something called a block copolymer, which is another fancy word. But basically the one that wants to aggregate at earlier temperature does, and it finds its neighbors, and then they make these spherical or cylindrical nanoparticles. And so we've used that to actually like package drugs, and use them as that's another way we've used them for drug delivery, is to actually put drugs in these little protein nanoparticles. Now, to your other question, you can put metal nanoparticles in there. You can make them conductive. You can put it's a pretty interesting field of research. People put iron nanoparticles inside, package them. So in this case, we would be packaging iron and iron nanoparticles in a larger protein nanoparticle. And use those to enhance like MRI contrast imaging, that type of thing. But it's totally feasible. Did I answer your question? Cool. I guess to talk about what Avi was mentioning earlier, when somebody is actually working for me, I'm trying to train them to be a graduate student and to think like a graduate student, and basically to think independently. So my like eureka moment with the undergrad that's working for me right now is when I went to talk to him about what he'd been doing the last week, and he told me all of the problems that he ran into and how he tried to solve all of them. And that second part is very critical. And that's not something you get to immediately. You should never be afraid to ask questions, but you should also be you should always try to answer the question first before you try to ask it, because oftentimes it's really something that you could solve, and actually the process of learning how to solve it is more of a learning experience than going to somebody and them telling you how to solve that problem. So that's a big thing that I look for is the ability to work independently and sort of analyze and answer questions on their own.
For the thing that you were using, like when you heat it, like you said that you could change like the temperature at which like it changes from a solid to liquid. So how would you like do that?
Yeah, so this goes back to that genetic specificity that we have. So certain different amino acids have different hydrophobicities. They have different ways that they like to interact with water. And so by changing some of the amino acid sequence, we can basically tune at what temperature it hates water the most, if that makes sense. So if we add a more hydrophilic or even a charged amino acid, we're going to increase that transition temperature because water likes to hydrate hydrophilic amino acids. If we add a more hydrophobic one, then it's going to want to solvate with itself more than water at a much lower thermodynamic peak or a much lower temperature. But good question. Anybody else? Anything about life? Just want to chat about philosophy. I can try that. All right, yeah. What do I plan to do after my PhD? So great question. There are a few routes for a PhD in biotech. One is, and the most traditional, is academia, and that would be going from a PhD and then to a postdoc. Hopefully just one postdoc. That would be 3 or 4 years, and then you would get hired as a faculty to do a tenure track faculty. One is to go into industry. That can be a small startup industry. It can be a large biotech firm. And the other, which is I think the route I'm going to try to do, is to start my own company. So the stuff that's part of the reason I wasn't able to talk about the stuff I'm working on right now is because we're actually going to try to start a company on it in the next year or so. So I'd like to do that, stay around, work with my boss, and get that up and going, and then I don't know what's going to happen after that. So I tend to plan it like 1 year at a time. So maybe like 1 day at a time sometimes. Yeah.
What would you recommend to major in? Like, what would you recommend for us to go into?
Yeah, it's a super variable question. The honest to goodness answer is whatever you're going to be most interested in. Like, I did BME undergrad, biomedical engineering, and I'm doing BME grad school. There's a lot of people that did mechie, electrical engineering, just straight biology, math, and actually a lot of those people have an advantage coming to a different major as a graduate program because they bring a whole different central set of skills to that they can apply in a new environment. So I mean, if you want to go into biotech, or then you probably got to do something in the sciences. But whether you're pure math, physics, you want to do engineering, all of those are very hireable people as long as you actually as long as you develop the skills in that field. So.
Oh, so
Yeah, I can just come back there and we can talk if you want.
Okay, so what was it like? I don't know if this is too personal, but like, what was like your GPA and like SAT and like your like student life, like, cause you're pretty successful, so like.
I guess I can answer that question, but I'm gonna say the caveat with that. I know a lot of people not in this, so I was basically in the upper 5 percentile for most of those things. My GPA was a little lower. I was around like a 3.85 or something. So I guess upper 10%. But my boss made a really big point when he hired me to be a PhD student. He doesn't like to hire people with 4.0s because he likes them to have a personality. So, and if you have a 4.0, you do have a personality, I promise. It doesn't really matter as long as you have a way to sell yourself. And those numbers really just get you in the door. But if you're persistent, if you have a strong personality, if you have a way to sell yourself, if you have a way to show that you're competent, then those numbers don't really mean anything. They're really just like a screening device. So. In other words, take those things seriously, but you can forget about them once they're over. So, also be persistent, by the way. I got one of the jobs that I had by literally calling the highest person whose phone number I could find, and every single week until they finally called me back, and it was like 8 weeks later, and they were like, "You're super nice, but why'd you call it so aggressively?" But just a way to get yourself in the door. So if someone tells you no, ask them why. Or if they ignore you, just keep asking until you get tired. So.
Yeah, I can vouch for that too. One of the big things for all things, because like I think I've told you guys before, I wasn't even in the top 10% of my high school class or even close to it, and I had the lowest SAT scores of anyone I met at WashU. But then at WashU, I was setting the curve, and I taught this genius over here. But one of the key things that I'm always have been able to get in is because I do have a personality, and I'm not afraid to contact people and to make those connections. And to most importantly, and this is the thing as I'm hearing about a lot of you guys in your interviewing process, is follow through and follow up.
Yeah, I have a fun story on that real quick. When I was applying to graduate programs, they tell you to email the professors that you're actually interested in at the program and tell them why you're interested in their research, because they're gonna be the ones that contact the what's it called? Admissions office and say, "Oh, can you actually pull that person's file? I'm interested in them." So I didn't do that for a while because I applied to like 8 or 9 schools and I was tired. And so when I finally did, it was only because I had a recent update to my resume. And so I called, or I sent an email to the current dean of graduate studies at Duke. It was like, "Hi, I have an update to my resume. Like, who do I contact about this?" And his response was, "Nobody really cares. Just contact the professor and like let them know in case they care." And then my response was, "All right, so you're actually the guy I'm interested in talking to. So like, let's get a conversation going." And that also prompted me to be like, "Crap, I actually need to email all these professors." And then my like number of responses I got like skyrocketed just after I started emailing professors directly rather than just waiting on the school to find me interesting. Sorry, you had a question in the middle? Awesome. Yeah.
Why do you like your job?
That's a great question. I love it because I get to do whatever I want. Like my day, if I want, I mean, this is an extreme version. If I want to stay at home for an entire week, then I can stay at home for an entire week. If I want to work for 12 hours a day, I can do that. If I don't want to do a project anymore, I don't have to. And it's like, it's so far removed, like a PhD is so far removed from what you traditionally think about with school, where someone gives you a thing to do, they want it done a certain way, and you do that and give it back to them, and then you measure your success based on how you did what they expected you to do. When you're in charge of your own project, you are setting your own expectations. And so there's some stress in that, because if you succeed, it's usually your fault. If you fail that day or that week, it's also usually your fault. But it's also extremely liberating to just be able to do what you want. It's great. It's part of the reason I don't want to go into industry immediately, or at least into an industry that's not based on something that I've been doing, is because I really like the ability to decide what I'm going to do over the next like couple of years. So that, and also super important, the people I work with are great. So I work in a lab that's super collaborative. Some of my best friends are in lab. And so I some days I look forward to going to work. Some days when I'm not, I get there and then I'm happy I did, because the people are there are really cool. So.
When did you kind of decide / know that you wanted to pursue a PhD?
I think I'm still doing that. There are good reasons to do a PhD and there are bad reasons to do a PhD. I got a little bit fortunate in that I didn't really know what I wanted to do, and I knew I didn't want a job. And so, in my idea, a PhD was the next level. I can work on an independent project. Like it'll be just more education. Don't go into a PhD because you want more education and you don't know what to do. That's not a good reason to do it. It happened to work out for me because I learned that I really liked that independence, and I really liked that ability to sort of grow it, grow in the way that I think. So, yeah. Also, anytime you do something in lab, the first time you do it, it's going to fail. I promise. If you ever do something the first time and it works, then you probably did something wrong, and you need to go back and figure out what you did wrong. Persistence is absolutely key, and research can move really slowly. It can take months before something actually starts working. So that's why it's important to like what you're thinking about and enjoy the process of thinking about it, because sometimes actually doing it is super frustrating.
So what's like the work schedule like? Like how much free time do you get? Does it vary, or do you have like a specific
So I'm the worst
schedule?
person as a student. My schedule is ridiculously variable. I don't like to wake up before 9:30, so I usually don't get into work until 10 or 10:30. But I usually stay at work until 7 or 8. But that doesn't happen every day. Sometimes I have to get there at 7 to start an animal experiment. Sometimes I don't get there I like to do writing at home, so sometimes I just stay at home and write, and then I'll go in and do experiments in the afternoon. I'm usually in at least one day every weekend for some period of time, unless I'm traveling. But I mean, I like it, so it's not that big of a deal. That's also part of the reason it's you gotta like what you're doing, because there's a demand on hours from a graduate student that doesn't necessarily exist in before you get to that stage.